cellular, they look like many cells living together, but still each cell takes care of its own well-being, right? So the body organization is yet cellular, which means that those cells are not able to make tissue, because a group of cells, how do we define a tissue? Do you know that, Ma? How do we define tissue in biology?
It's a group of cells which come together to organize things, to do functions.
Exactly. Yes, exactly. It's a group of cells which come together to achieve a common function, a common goal. You know, so unless and until they do not achieve a common goal, or they do not function towards something in a coherent manner, they cannot be called a tissue. Even if they are loosely tied together, they will always be called cellular organization, because they are not working towards a common goal. Each cell is working towards its own existence independently. Okay, so this happens in porifera, which is in the kingdom Protista, and also it happens in slime mold. Okay, now slime molds that we are going to talk about are of two types. One is called plasmodial slime mold or cellular slime mold, but I think you don't have to go into that detail. So, but let me tell you, plasmodial slime mold and cellular slime mold. Now, what happens in slime mold is, with respect to their reproduction, you can make this one figure that will clarify everything. So it all begins with, let's start with a slime mold. So let's say this is a slime. So slime mold has no shape, right? It's like a slime. And you will find sometimes when they are developing, they even have gaps in between. So they are like slimy, gooey substance, which behave like an organism. Okay. So let's say this is slime mold. Mature slime. Okay. Now, when it matures under favorable condition, it prepares to fruit. Okay. It's called the fruiting part. Now, what it is fruiting is that it develops into, so it still stays like this slimy substance, but it starts developing these kind of structures on itself. You understand?
Yes.
Yeah. So this is called, this thing here is called sporangium. And if it is sporangium, what it is containing inside?
Spores.
Exactly. So if I make a sporangium, let's take this one. So I have this big structure here. So it has a stalk and a sac-like structure in which there are multiple spores. There are multiple spores present. Okay. Now these spores, this sporangium undergoes meiosis, forms spores through the process of meiosis. So here meiosis is happening. This is, you have to remember. So they do meiosis and they release spores. So let's say these are, make it black. These are spores. Now, what is the, if you have studied in class 10th, what is the difference between spores and seeds? Are they seeds?
No.
Can I call them seeds?
No.
No, I cannot call them seeds. So what is the difference between spores and seeds?
can be sown, whereas spores cannot be sown.
So they behave like a seed when they fall on the ground, they germinate. Okay, but spores are haploid. N means one copy of the genome. I told you about haploid and diploid in the last class, right, Maha?
Yes.
Seeds, on the other hand, are diploid, and seeds contain two things. One is the embryo and the other is endosperm. I also told you about endosperm, which is a triploid tissue. Yes?
Yes.
Yeah. Now, from here, it gets a little complicated. So if you pay attention, now I'm just removing this part here to complete it. Now these spores, they start germinating. So let's say this is a spore, and when it starts germinating, a new cell, like slime mold kind of a cell, comes out of it, and it forms, it is called amoeboid cell, because it looks like a, it has no shape. Okay, just like the mature parent from where we begin. Or the other way it can go is, it can grow into a shaped cell. So the shaped cell will have flagella. Okay, is it clear? This is called flagellated cell. So either it can be amoeboid cell, or it can grow into flagellated cell. Is it clear? And these two conditions are interchangeable. This is the complexity. So from the spore, so we call this thing as germinating spore. Is this clear till here? Parent,
Yes.
under favorable conditions, start making sporangium. Inside the sporangium, with the help of by the meiosis process. Meiosis we will study today. Okay, meiosis is reductional division. So this body is diploid, and it forms haploid. Stalk is diploid. Okay, it forms haploid spores. Now these spores, when they germinate, they can either become flagellated or amoeboid cells, and ultimately both of these, flagellated and amoeboid, undergo syngamy, fertilization. So half of the cycle is spore forming, and the other half is syngamy, fusion of the two. And as a result, a zygote is formed. Okay, now this is a zygote, a slime mold zygote. And zygotes are always diploid. So half of the cycle is in spore form, it's haploid. Then when the two different kind of cells fuse together to form a zygote, it becomes diploid. I also have to delete maybe this part. And then what happens? This zygote, which is 2n, then develops into slime, again, a slime mold kind of a cell, which is called plasmodial slime, also known as feeding slime. It eats a lot. It keeps eating, and then it becomes a mature slime mold. It is, and this is also known as mature plasmodial. Is it clear? Or a mature slime mold.
Yes.
Yeah. So this is how they reproduce. So you have to remember, you can write down a few points if you want. Under favorable conditions, so this part from here to here, this part is happening under favorable conditions. Write down under favorable condition, slime molds reproduce by, under favorable condition, slime molds reproduce by forming a reproductive stalk, this thing containing spores. Okay, these stalks release the spores which germinate into new mold cells. So either it can be flagellated or amoeboid. And then the other part, this is the spore formation part. The other part is reproductive part. Some molds also do one thing. So right from these spores, let me use a green color here. So right from the spore stage, they go directly into this stage. You understand? They skip the not sexual, but they skip this syngamy part completely. Do you understand?
Yes.
So this is how a slime mold reproduces. Is it clear?
Yes, but is from every sporangium only one zygote is produced?
No, sporangium is a broader term. Sporangium is which contains all the spores. From one spore, this is a single germinating spore. From one spore, either one kind of cell can be produced. So from one sporangium, thousands of spores can be produced. Many of these spores do not go this pathway, but can directly germinate to form a new mature spore. See, this is coming out of the sporangium, spore, sorry. Can you see something comes out of the spore? It either become an amoeboid cell, and this amoeboid cell can directly develop into this. You understand? It can just grow big and can develop into this sometimes. But sometimes it undergoes syngamy with the flagellated cell as well and become a zygote and then a 2N slime mold and then come to this part. So either it can be haploid or diploid depending on which pathway it took to reproduce. Okay. So all this part from zygote onwards is diploid till sporangium, and from spore to syngamy is haploid. Clear?
Yes.
From one sporangium, it's not like only one. What did you ask? One
zygote.
No, it's not. There can be hundreds. Okay. But mostly they in the favorable environment, they just go through the easier way, and they are called the term plasmodial is for the slime molds, is for given to just differentiate them from the spores, like the spores and the flagellated part of their life cycle. Okay. And if you come to the slime molds live in what do you say, damp areas, right, where the woods are rotting, because they are kind of protists that depend on, they are saprophytic, right?
Yes.
And in classification, it comes in kingdom Protista. Okay. And slime mold contains more than 700 different species. So it's not like all slime molds behave the same. But you have to know that the one that is that we are studying and is very important is myxomycetes. One example is myxomycetes that is asked. Just remember this one. Mostly this term mycetes you will find for fungi, deuteromycetes, etc. In your textbook in chapter 2, go to the open the fungi part, fungi kingdom.
Yes.
Open. What can you see in fungi kingdom?
Phycomycetes, ascomycetes.
Yes. So you read this word mycetes.
Yes.
But remember, myxomycetes are protists. They belong to the slime mold. Okay. Fungi are multicellular and they behave like they can have loose tissue organization. Actually, if you go into the biology of both slime molds and fungi, it's not very different. Both are saprophytic, both feed on dead and decaying matter. Both are heterotrophic. But point is that slime molds can operate through spores. Even fungi has fungal spores are there. You know that? Fungi also has this sporangium.
Yes.
Thing. Yeah. So that's where for many organisms, slime molds, and there are many others for which this classification becomes very fine-tuned. If you ask me why are they in kingdom Protista and not in kingdom fungi, I can say two things. First, they do not have a cell wall made up of chitin, and second, they cannot show loose tissue organization. They always show cellular organization. Okay, and as for the nutrition, heterotrophic, and they basically do phagocytosis if they find small food like amoeba. Amoeba can do phagocytosis, correct?
Yes.
So they also similarly can just eat anything up by extending pseudopodia. And amoeba is a protist. You know that? Amoeba comes under kingdom Protista. So all these behaviors make slime molds also come in kingdom Protista. Okay. But they are very much like fungi as well in terms of their feeding and habitat, especially habitat. Amoeba and fungi live in very different habitats, but slime molds and fungi live in similar habitats: damp, decaying, rotting woods, etc. Okay, is it clear?
Yes.
Any other doubt you have from chapter?
Yes. Yeah. And kingdom fungi, basidomycetes, how do they reproduce?
How do deuteromycetes reproduce?
No, basidomycetes.
Basidiomycetes.
Yes.
Okay. So do you understood the difference between phycomycetes, ascomycetes, and deuteromycetes and basidiomycetes?
Yes.
Like the difference is basically on the basis of what?
I guess mycelium.
One is mycelium, yes. So the difference, the difference is also on the basis of how they reproduce. You know that?
Oh, yes.
For example, the word deutero. Deutero word is for false. Okay, like which fake something. So deuteromycetes are called imperfect fungi because they only have asexual reproduction. Fungi have both life cycles, through spore formation and through sexual reproduction as well. Now, in basidiomycetes, the asexual spores are not found. Okay. So how they reproduce is that the mycelium is branched, okay, and asexual spores, the same thing that you are seeing here in myxomycetes, which is a slime mold. There is an asexual spore stage that is missing in basidiomycetes. So they have two options. Either reproduce vegetatively. If they have to reproduce asexually, they go through vegetative reproduction. First is fragmentation. So you can write down for basidiomycetes. This was your question, right?
Yes.
So no asexual spores. So they do not reproduce through spores. But instead, they have vegetative propagation or vegetative reproduction. And you have studied in asexual mode something called fragmentation. Remember chapter?
Yes.
Yeah, fragmentation. What is fragmentation? So what happens is these basidiomycetes, it's like a fungi which grows, and when it breaks, so this new part falls on the ground. Okay, this falls on the ground and starts developing into a new basidiomycetes. So they can develop from their fragments. You understand? That's fragmentation. That is first way through which they can reproduce. Okay. Second is plasmogamy. So they do not have sex organs, like they do not produce cells here differently like a flagellated cell or amoeboid cell. But what they can do is they can fuse their plasmogamy. This is the word. So one is this. The second is they reproduce through, write down, plasmogamy. Now, tell me, you have studied it. What does the word plasma means in biology?
Fluid.
Yes. So their hyphae or mycelium must be having some fluid filled inside, right?
Yes.
So, and gamy means, always write down, fusion or union. So syngamy is where two cells fuse together. Here also the word gamy is there, right? Here also the word gamy is there. In this case, what they do is they fuse the fluid present in two different cells or two different, what can you call, mycelium. Okay, and this way they can produce something known as basidiospores. So they reproduce either through basidiospores, second, or through fragmentation. And to make basidiospores, they have to depend on plasmogamy. Is it clear?
Yes.
And the different thing about basidiospores is it is externally formed. It is not formed inside something. It is externally produced. And there is a structure called basidium. Okay. Or basidiocarps. It is produced there. Clear?
Yes.
Ma? Yep. Is this drawn?
Yes.
Yeah. Do you have any other doubts from any other kingdom? If you have, like, we can, if you need, we can give you the same kind of extra classes next week as well. Okay.
Oh, that's
It's okay. Like,
Yeah,
is
it's okay.
chapter 1 and 2 is done for you, right?
Yes.
So can we now, because I will again come to chapter 1, 2 and things. So for now, let's go with chapter like 10 and then we'll come back to it.
Okay.
If you feel like there is a need, you can let me know or let the team know. Okay.
Excellent.
Okay. So we are joined by Iram. Hi, Iram. How are you?
Hello, sir.
How are you?
Yeah, I'm well good. How are you, sir?
Very well. Can you see the screen that I'm sharing, everyone? Both of you? Yep. Okay. So what about Aisha? Is she joining? Iram?
Yeah. That's right.
Okay. So let me wait for her to join and I'll be back in a minute. Okay. Who will give a brief summary today? Ma, can we go with you?
Okay.
Okay, good. Iram is very, very supportive. She helps everyone who gives summary. She helps me when I give summary. So I have not given one. Yes, see, there's a thumbs up already. Okay, so I'll be back in a minute. We'll start when Aisha joins. Okay, Iram, can you contact her and let her know that?
Yes, sir. Yes.
Yeah, please. Okay, everyone. Aisha has joined. Hello, Aisha. How are you? We were waiting for you to join. Am I audible?
Yes, sir.
Yes. Good evening, Aisha. So, yes. So let's go to Maha for a brief summary of the last class, and Iram and Aisha will help her if she finds difficulty somewhere. Okay, and I'm also here. Okay, Maha, over to you. Are you there?
Yes, sir.
Yes. Let's start. So, what we did in the last class? Quick.
So we learned about the cell cycle and the phases that it goes through when it's going to divide. And we learned about the interphase and the M phase, and that the M phase takes about like 5% of the cell cycle, whereas the interphase takes about like 95%.
Mhm.
Yeah, we learned about the G1 phase, S phase, and G2 phase, and about the
And
G0 phase.
what are those phases? What are they known as?
The growth phase, the synthesis phase, and the growth phase too.
Yeah, it can also known as the gap phase. Okay. Yeah.
Yeah.
Good.
Yeah, and the G0 phase, which the cell does not go through any division.
Mhm.
And then we started with the prophase, and yeah, we did like half of the prophase or something.
Yeah, we also learned that in M phase starts with karyokinesis, right?
Yes.
Yeah, and what was that? Iram, would you like to come in and help? What was M phase?
Yes, sir.
Yeah.
M phase is also called as the mitotic phase. It's
Mhm.
It consists of two phases, karyokinesis and cytokinesis. Karyokinesis is the division of nucleus. There are
Yeah.
4 major phases of karyokinesis. There is prophase, metaphase, anaphase, and telophase. After these phases, there is cytokinesis, which is the division of cytoplasm.
Yes, right.
We have
So,
prophase.
before going to prophase, let's talk about interphase. Okay. Because prophase comes in M phase, right? We have to do interphase and G0 phase first. Iram. If you're speaking, you're mute, Iram.
First is our G1 phase, which is also called as gap 1. It is the longest phase, and
Yes.
it is between M phase and the M phase and the S phase, and DNA replication growth occurs in this phase. Second is the S
No.
phase.
In G1 phase, there is no DNA replication. G1 phase is between M phase and the DNA replication. Okay. There is growth, but there is no synthesis in this phase. This is what makes G1 different from G2. In G2, there is growth with synthesis
So,
of proteins.
isn't synthesis of protein and RNA synthesis occur in G1 phase?
In G1 phase, some RNA can get synthesized, and the proteins which are normally required for the cell, when cell is living, right? So cell is, in a way, cell is always synthesizing something or the other. It is synthesizing and breaking down something or the other all the time. But this synthesis, we are talking in terms of cell division.
Okay.
So the synthesis of proteins, which is done in G2 phase, is the proteins required for division. See, I have mentioned it here. I'm not talking about housekeeping proteins or normal proteins that are required every time, because without synthesizing something, cell cannot survive. You understand? But here it is with respect to division. So in G1 phase, no DNA replication happens. There is growth, but the synthesis does not happen. Active synthesis for division. The synthesis for survival, of course, that is there in every cell. Okay, but from cell cycle point of view. You understood, Iram, Aisha, and Maha?
Yes, sir.
Yeah. Yes, sir. Yes, Iram, continue.
The next is S.
You got mute,
The
Iram.
next, yeah, sorry, sir. The next is the S phase, which is the synthesis phase. Here, the nucleus divides, DNA replication occurs,
Yes. Now,
and
just to prove your point, just one second, Iram. So in S phase, it's not like just the DNA synthesis happens. You know, every protein synthesis is happening, RNA synthesis is happening, but we call it S phase from the context of the cell division cycle. So the DNA replication, which is a crucial event, happens in the S phase. You understand? Iram, there?
yes.
Yes. Continue.
And also the centriole duplication also takes place in the S phase. Now,
Yes. Right.
G2 phase, it is the cell continues and cell growth continues as well as cell synthesis, and it is mainly proteins and proteins are synthesized in these phase G2.
Proteins required for division. Don't forget that.
Then there is G0 phase as well. If the cell do not wants to divide, some cells that do not divide undergo division, they enter the G0 phase, and these cells are usually neurons, cardiac cells, and they are metabolically active. And this stage is also
Yes.
called as quiescent
But
stage.
why we call them inactive? Because inactive cell cycle, from the perspective of cell cycle, they are inactive, but metabolically they are active. Yes. Then we discussed about ploidy. Let me go to Aisha for this part. Aisha, would you like to explain ploidy or discuss ploidy? I'll be with you. Come on, try. Yes.
Ploidy means a copy of genome.
Yes.
And ploidy is divided into haploid, diploid, and polyploid.
Mhm.
Haploid means they have one set of genome. For example, gametes of plants. Then we have diploid, which has two set of genome.
Yes.
And animals are mostly diploids. And also plants. Then we have polyploid. They have 3, 4, 5 sets of genome, etc.
Exactly.
And plants can tolerate polyploid. Also, endosperm is a triploid tissue in plants.
Yes, right. So basically, in animals, mitosis is observed in diploid somatic cells. This is why I was explaining this. And in plants, mitosis is observed both in haploid and diploid cells. So this is the thing that you have to remember from this part. Okay. Then we started with the M phase or the mitotic cell division. Yes, back to Maha. Maha.
In mitosis, the ploidy of the parent cell and the daughter cell remains the same. Therefore, the
Yes.
mitosis is also known as equational division.
Yes.
4 stages of cardiokinesis: prophase, metaphase, anaphase, telophase.
Yes, and I told you that the analogy that you all are here to celebrate a ceremony. So this is the ceremony starting, right? From prophase. What happens in the ceremony, I think we finished with prophase only. Yes, so tell me prophase. Then I'll start with metaphase. Iram.
Just a minute, sir.
Yeah, sure.
Yes, sir. In the prophase, it's the first phase of mitosis. Here, the cell organelles will disappear, the nuclear membrane disappears, and the condensation of chromatin will start appearing in the form of chromosomes. They're tightly, compactly packed.
Yeah, the X shape will start coming.
And centrosomes that got duplicated during the S phase in cytoplasm begins to move toward the opposite poles of the cell.
Yes.
And at
And
the
then
end of prophase, Golgi complex, ER, and nuclear membrane will not be visible.
so basically all these will just go away because we do not need endomembrane system right now. We need the cell to divide. So for that we have to get rid of the endomembrane system so that the cell can divide without any problem. And once it is divided and the new cell again enters into the growth phase G1, then everything is made again and it comes up. You understand? Too much of hard work, right? Okay, so prophase is where things started. And now let's go to the second phase, which is metaphase. Now, as the word says, metaphase. In metaphase, by the time a cell enters the metaphase, now again, let me tell you that when it is happening, it's not like after every phase there is an alarm or something that goes up and we can distinguish. We just make these demarcations on the basis of some characteristics. Now for metaphase, by the time the cell enters the metaphase, the nuclear envelope is completely gone. It's completely disintegrated and is gone, because now the most important thing, the characteristic feature for metaphase, write down. Characteristic features. So for every phase, you have to remember something. For prophase, the chromosomal materials start to condense, means both the the parties, the bride and the groom, starts packing their stuff. So they are becoming compact. And the centrosome in the cytoplasm, so in the nucleoplasm, this is happening. And in the cytoplasm, centrosomes are moving towards opposite poles. In metaphase, the characteristic feature is that the nuclear membrane is completely gone. And remember the compact chromosomes, their packing is finished. Now they will start moving towards the central plate. Okay. Now write down the first thing, the nuclear membrane completely disintegrates. The nuclear membrane completely disintegrates. And the chromosomes are spread out in the cytoplasm. Spread out in the cytoplasm. At the beginning of metaphase. Now metaphase, the most important thing that happens is formation of metaphase plate. Okay. So remember that the chromosomes are made up of two chromatids, the X shape. Just to revise, let's go up. Where did we make the chromosome? Oh, we have not made the chromosome. Okay. Did I tell you about the X-shaped chromosome? So this X shape is because there are two chromatids, right? One, this is chromatid 1 and this is chromatid 2. They are called sister chromatids. Yes or no?
Yes.
Which gives rise to one chromosome.
Yes.
So at metaphase, the sister chromatids come together and align at the metaphase. Okay. So write down. At this stage, the chromosome is made up of two sister chromatids. At this stage, the chromosomes are made up of two sister chromatids joined together by what keeps them together? What is in the center, this blue thing?
Centrosome.
Not centrosome. If you write centrosome, now you will get a big 0.
Oh yeah.
Centromere.
Yes. Centrosome is what was moving towards opposite poles. Okay. Centromere is what keeps these two sister chromatids together, because ultimately these sister chromatids are going to be separated. Okay. Understood, everyone?
Yes.
Yeah. So held together or joined together by the centromere. And write down 2 small discs. Now I'm making it big so that you can see, but they are small. 2 small discs are at the surface of the centromeres, known as kinetochores. These are proteins, basically protein discs. 2 small discs or 2 small disc structures are present on the surface of the centromere called kinetochores. And about kinetochores, the question is, write down, these are the site of attachment of what? What is coming to attach to them, to pull them apart?
Spindle fibers.
Exactly. Sites of attachment of spindle fibers. And who will throw out the spindle fibers? Who's the Spider-Man throwing this web out? What was moving towards 2 opposite poles?
Centrosome.
Yes, yes, centrioles in the centrosomes, right? Very good. So write down. What have you written? Attachment sites for spindle fibers, right? Write down. The metaphase is characterized by. The metaphase is characterized by all the chromosomes coming to the equator or center of the cell. All the chromosomes coming to the center of the cell with one chromatid. So there are 2 sister chromatids. So with one chromatid attached to the spindle fiber from one pole. One chromatid attached to spindle fiber from one pole, and its sister chromatid attached to the spindle fiber from another pole. That's why there are 2 kinetochores. Okay. Is this clear?
Yes.
So pay attention to the details here, because this is what, like, this is the area for questions. And this is known as the metaphase plate. So this is referred to as formation of metaphase plate. Okay, and it looks something like this. So if I draw it for you, let's say this is a cell. Very bad. Cell made. let's say if this is a cell, so it has one spindle fiber here and the other centrosome here. So spindle fiber goes in all directions. Okay, don't just think that it goes in this direction only, because but in this direction there is not much space to go, right? So it just reaches the end of the cell and stops. In this direction already, how should I make it? So this is one and this is other chromatid. This is one and this is other chromatid. You understand? These are two sister chromatids. And from each pole, it goes and binds to one chromatid. From here also, it goes and binds to the other chromatid. You understand what's happening? Towards both the sides, there are kinetochores on the centromere. Do you understand, everyone? And now when they will be pulled apart, the sister chromatids will separate. So this is called metaphase plate. This, oh, so bad with these things sometimes. This part. Can you see that it's like a plate? This is called metaphase plate. Is this clear? So if I ask you what are the two characteristic features of metaphase plate? So one is spindle fibers attached to kinetochores, spindle fibers from both the ends attached to kinetochores of the chromosome. And second, chromosomes are moved to the equator, the center, and get aligned as a metaphase plate. Okay. You understand? Yes, no, something will help.
Yes.
Yeah. Then now comes anaphase. What you have to remember here for your NEET exams is that anaphase and telophase, they are difficult to distinguish, but they are done on this basis. So anaphase can be early anaphase or late anaphase. Late anaphase looks like telophase, but without going into the complexity of that, just know that at the anaphase begins when these spindle fibers begin to shorten up and start pulling these chromosomes apart. Okay, so write down. At the beginning of anaphase, the chromosomes that were arranged at the metaphase at the beginning of anaphase, chromosomes arranged at the metaphase plate begins to split apart. Begins to split apart. And from here onwards, the moment they have split, we no longer call these chromatids as sister chromatids. We call them as daughter chromatids. Okay. Do you understand, everyone? So this is the thing that you have to remember. In anaphase, the moment these two sister chromatids separate, so these two here are sister chromatids because they are joined together by a common centromere. The moment they split apart, they do not have common centromere any longer. Is it clear? So now they are known as daughter chromatids. Write down. The daughter chromatids which will further be known as will become daughter chromosome. So daughter chromatids is daughter chromosome, no difference. It's just a single chromatid chromosome, not the X one, but the single chromosome. And later on, it will be called the daughter nuclei. When the envelope will come back again, it will be known as daughter nuclei. So these, the daughter chromatids, begins their journey or migration towards the opposite poles. This is karyokinesis in action. Understood? Yes or no? And one detailed question that has come in exam is about the arrangement. So again, here, this is the centrosomes. And these are the spindle fibers they have thrown. They're also on the other side. Now, one thing that you have to remember, when they are moving, they are literally like, so if I hold you by your hand and start pulling you with speed, what will be the first thing that will be in the leading direction of your motion? If I'm holding your hand and I'm pulling you literally with force towards a pole where I am standing. So what part of your body will be in the leading direction, and what will be trailing? Yeah, the hand will be leading, right? And rest of the body will be trailing, correct? It's the same. So what part do you think will be leading in this case? What it was attached to?
Kinetochore.
Kinetochores.
Yes. And where are kinetochores? Kinetochores are just disc-like structures on
Centromere.
Exactly. So the centromere, which is now split, right? Centromere is divided into two. This will be like this, and the single chromatin will now be, you will see it like this, literally trailing, right? You understand?
Yes.
This was one detailed question which has come in this. In anaphase, the leading part of the chromosome towards the pole is short arm of the chromosome, long arm of the chromosome, centromere. So the answer is centromere, okay? Just mark it. This is centromere, which is leading. And this is the chromatid, which is lagging. Or trailing behind. So if I ask about the two features of, the question is two features of anaphase, you just have to say one is centromere split, chromatids separate, and then chromatids move towards opposite poles with centromere leading and the chromatids trailing. Clear?
Yes.
Yeah. Now, in the end, the last part of karyokinesis is telophase. Now in telophase, chromosomes finally reach till this very end here, where the centrosomes are present. Okay, and the moment they reach their opposite poles, now they are in a region where finally everything is done, so that one event, that transient event where the ceremony, the union was being done, was this at Metaphase Plate. And it was a very short-lived event, right? As I told you, in the whole marriage ceremony, the actual thing is very short-lived, right? It's very short ceremony. But most of the time came in transporting it towards the Metaphase Plate, then taking it to different directions. Once they reach their respective poles, they start to what? Decondense. You know, it's just like once the ceremony is done, you're back at home again, you are in your own space, then you will again decondense, right? You will not be compact. You will take off jewelry, your purse and everything, and you will keep it at random places. Yes or no? You understand, everyone?
Yes.
So write down, in telophase, chromosome clusters reach the respective poles. Chromosome clusters reach their respective poles and begins to decondense. Begins to decondense. It also, in some books, it's written as their individual identity is lost. They will open up like threads. Their individual identities are lost. So if you stain the chromosome, you will see it till the late anaphase, but in the telophase, you will not be able to see the chromosomes. Okay. And the second thing, why are they being very comfortable and cozy now? Why are they decondensing? Because nuclear envelope has started coming back, right? So write down, the nuclear envelope forms back. Forms back around the chromosome clusters. We know that they are there, right? It's not like they disappeared. We just cannot see it as chromosomes, compact form, stained. They just disappear when we see it under the microscope. But they are there, they are just decondensed, and the nuclear envelope form back around the chromosomes at each pole. So now there are two nucleus, right? And they are called to form two daughter nuclei. To form two daughter nuclei. So two daughter nuclei in one cell, but they are still present in the single cell only. This was the cell. You will start seeing. So at the very end will be your centrosomes, but all your threads have reached here. Similarly, all your chromosomes have reached here. So nuclear membrane will start developing around it. Okay, at both the ends. So these are daughter nuclei. Okay. And also what happens? You will see that there is a small furrow formation that has started in the cell, because this is marking the initiation of cytokinesis, but still karyokinesis is finishing. But the third thing that happens, write down, nucleolus, Golgi complex, and ER. Nucleolus, Golgi complex, and ER begin to form. And initiation of cell division furrow can be seen. This is the furrow. And you will see that these type of structures will start appearing on both the sides, ER, Golgi, and the cell finally will divide from the center. Karyokinesis is finished. Now the last part is cytokinesis. After the telophase, it is cytokinesis, right? Write down. Cytokinesis is the division of the cell itself after karyokinesis resulting in two daughter cells. Okay. Now this cytokinesis, now we have to see, till the time things were happening inside the cell, they were happening the same for plants and animals. Yeah, you understand?
Yes.
Okay, but now when the cell has to divide, what is the basic difference in a plant and an animal cell? The first major basic difference.
Plants are composed of cell wall.
Exactly, perfect. Plant cells have an additional layer called cell wall. And what are the properties of cell wall? What is cell wall? How is it different from cell membrane or plasma membrane?
Cell wall is rigid.
Cell wall is rigid, right? And plasma membrane is fluid, as I have told you, fluid mosaic model, right? So yes. Plasma membrane is not a problem. You can easily pinch plasma membrane. So that's why in animals, cytokinesis is very simple. There is a furrow. Okay. Actually, just write down furrow is seen in animal cells. So in animal cells, it's very simple. You make a furrow, and then that furrow will keep deepening and keep going into and into until it pinches to the cell into two halves, and it's done. Simple. But in plant cells, it's slightly complicated because of the presence of cell wall. Okay. You understand? It is difficult to pinch a cell wall from outside, because that is the whole purpose of cell wall, no? To provide rigidity and support It's like the walls of our house. You cannot pinch it. So biology is very logical. If there is a problem, always try to extrapolate that problem on your real world. So if you are living in a, let's say in a plastic tent, okay, you're going on a picnic, hiking, and you're living in a plastic tent, which is a very big tent. So to divide it into two, it's very simple. You take the top of the tent and just from the center, bring it all the way down to the ground, and then there'll be two different compartments formed. Yes or no? Aisha, Iram, Maha? This is animal cell dividing. But let me ask you, if you have to divide a big room into two parts, which is made up of concrete walls, how will you do it? Will it be able for you to pinch the walls now, bring it closer and divide the space into two?
No.
Then what will you do? Iram, what will you do, Iram? As a, let's say you are a construction worker and you have been given this task to divide a rigid space, a rigid room containing walls into two rooms. Yes, how will you do? And you cannot quit, huh?
I
If you are
cannot
thinking-
certainly do it.
You cannot? You cannot what?
No, sir, it won't be possible.
So which means once when, yes, Aisha,
We can construct a wall,
we can construct a wall where?
the middle of the room.
In the middle of the room. See, Aisha has a way. Iram thought, okay, once a room is made, a room is made. But Aisha, correct, very good. But if you are constructing a room in the middle, sorry, constructing a wall in the middle of the room, will it be outside in or inside out? Like you are doing it from inside the space, right? Already
Yes.
within the room, right? When we were pinching that tent made up of plastic or any fabric, we were doing it outside in. We were taking the surface, the top surface, the cell membrane, and we were bringing it to the ground like a furrow in animal cell. Yes.
Yes.
But when you are making a wall to separate two space in a room, you are making a wall already inside the room. Yes or no?
Yes.
So just because this is the only logical way to solve this problem, biology solves it the same way. Okay. In case of animals, it pinches the plasma membrane. Yes, Aisha, it is. In case of animals, it pinches the plasma membrane outside in to make two different, just like you can pinch a balloon to make two different small compartments. But in case of plant cell, it starts making the cell wall in the center of the cell, which grows towards the outside and then gets connected with the cell wall. And hence there are two different cells produced. This is how cytokinesis happens. So cytokinesis in plants and animals happens slightly different, just as the logic suggests. You liked it or not? Biology is not just anything happening. It's like you thinking and doing something. Even, the biological phenomena is also thinking and doing it. Oh, it's not possible to pinch a cell wall, so let me make a wall from the center. Okay, so write down. In animal cell, Hiram is very happy. Something that we do, cells are also doing. Cells are, so you can just imagine yourself as one cell when you are behaving in the outer world. Every cell is doing the same thing inside your body, basically. Yeah. Neurons behave the same. Yes. So if you want to be more stronger in society, in your school, in a group of people, what do? Who's called a better, like a more stronger person in society? Who's well connected, right? Yes or no?
Yes.
If in a school you have you have more friends, less or no enemies, everyone thinks good about you, everyone is a friend, then you are a more accepted person in the society or in the college, and then probably you have more chances to run for the college president or school head boy, head girl, right? You are more popular, basically. Yes or no? So you are a stronger person in the society. Similarly, neurons which make more connections with other neurons are stronger neurons. You know, they help us more than neurons who have less connections, because they will over time be fade away. So all our stronger skills, like if you are good at, like I think we have discussed it, have we in this class, that our abilities are shaped by the connections. Did I tell you about that brain is like a forest? No. No? Okay. Let me tell you this very interesting thing. Brain, all the neuron connections in the brain can be thought of like a forest. So in the forest, let's say, have you seen the paths which are traveled by more people? So when you walk in a forest, you walk in a line, one behind the other, right? For your own safety. You will not walk like a group of friends going to McDonald's or something, right? You will be aware, and they always walk in a line, so a track gets created. The most traveled ones have no grasses in that track. You understand?
Yes.
And they are visible in the grass. So that's called strengthened tracks. So the more you walk on it, the more it gets strengthened. Similarly, the connections which are used more in our brain get strengthened over time. And neurons, which make more connections with nearby neurons, are the ones playing important role in our memory, processing of the information, making us learn certain things like that. So it's similar. One neuron is behaving like the same. See, if I want to take up a major role, if I want to give this person some good abilities, let me make more connections. Same thing you do in your real world. Yes. So if you learn biology through these analogies, extrapolating it on something or the other, it becomes easier. Then you don't have to remember certain definitions and terms. Yes or no? Cool. Very good. So come to cytokinesis, write down in animal cells. In animal cells, first point. In animal cells, this is cell division is achieved by, in animal cells, cell division is achieved by forming a furrow in the plasma membrane, forming a furrow in the plasma membrane. Second point, this furrow gradually deepens into the cytoplasm. This furrow gradually deepens into the cytoplasm and ultimately joins in the center, because the furrows are coming from two opposite sides, right? And ultimately joins in the center to divide the cell into two parts. These are the two daughter cells, okay?
So,
Yes, yes, Ira.
so bacteria also consist of cell wall, right? Also the
Yes.
division of plants.
Yeah, so in bacterial division, in bacteria fusion, it's again, when this happens, so it also has a cell wall. So sometimes what happens is bacteria have two different ways. One, under unfavorable conditions, sometimes it creates a cyst around it, a very, very impermeable tough covering in which nothing can happen. Okay, so in order to get out of it, it has to dissolve certain parts of the cell wall. Whereas normally when a bacterial cell divides, you're right. So there also, because there is a cell wall, peptidoglycan layer, but that cell wall can be formed on the outer side of the membrane only. Remember, there were polysaccharides also. So there what happens, the membrane divides and then the cell wall is formed in prokaryotes. You understand? So it's not like plant cell. Plant cell wall is made up of cellulose. Cellulose is just a polymer sheet. Cellulose is basically carbohydrate only. Starch and cellulose are two things. We can digest the starch part, and which is very less in leaves. That's why we do not eat grass. Even if we eat grass, we will not be able to get nutrition from it because cellulose we will not be able to digest. We do not have the enzyme called cellulase that is present in cows, goats, and all the cattle that graze on grass. So cellulose is a tough polymer which cannot be broken easily. So that's why this pinching is not possible. So it's inside out wall formation. Whereas in bacteria, depends on what kind of bacteria it is. So it can be gram negative, gram positive. It also depends on the thickness of the cell wall around, and etc. Okay, good question though. But it's different in bacteria than plants. Plants are eukaryotes also. Bacteria are prokaryotes. So that also makes it different. Now write down in plant cells. In plant cells, the concept of mitosis, meiosis is not there in bacteria, okay? Because they have just one DNA thread. And that is also circular in most of the cases. Prokaryotes have circular. Remember, we did the prokaryotic versus eukaryotic cell. They have a circular DNA.
Yes.
Yeah. So their things are slightly different. We'll come to it later. Like I think in some of the other chapter, we have to study that. Now write down in plants. The cell wall in plants is inextensible. It cannot be extended or pinched. It is rigid. The cell wall in plants is relative to the cell membrane. Inextensible. It cannot be extended. Therefore, they undergo cytokinesis. They undergo cytokinesis in an inside-out manner. In an inside-out manner. In animals, it is outside-in manner. Okay. Let me just write it here.
So their karyokinesis is same as animals?
Yes, karyokinesis, because see, till the time something is happening inside the cell, it does not matter your cell is layered by one cell wall or 20 different layers. All is happening inside, right? Nuclear membrane will dissolve and things will move to opposite poles. That is similar. That is same as animal cells. Okay, in plant cells as well.
Okay.
But when it comes to the when it comes to the cytokinesis, then what the outer layer matters. So what is on the outer side matters a lot. And here on the outer side is a cell wall, which is inextensible. So they have to go in an inside out manner for cytokinesis. Okay. Write down. The wall formation starts in the center of the cell. The wall formation starts in the center of the cell. And grows outward. And grows outwards to join the existing lateral walls. Okay. And this cell wall and this wall formation, write down, this wall formation begins from a cell plate. This wall formation begins from a cell plate. Let me draw it to explain it to you better. So let's say this is a plant cell. So plant cells have rigid shapes, right? Because of cell wall. Yes? Yes. So this is the cell wall. And here, inside, in the center, a thing grows, which is known as the cell plate. After the two different nucleus has been produced here and here, these are daughter nuclei with their chromosomes. So this cell plate then starts growing out and meets this already present lateral cell wall. So when it meets here, after this, these two cells are called separate cells. But you know, we studied one more thing. I'll now ask Iram to shed light on this. Iram, do you remember something called Plasmodesmata? We did it in the last chapter. Can you go back and tell me what was Plasmodesmata?
It is the I don't exactly remember the definition.
Yeah, go to the notes. That's why I'm saying go to the notes. Aisha, you can also do it. Whoever finds first can shed some light on this.
Yes, sir. It is the connection between two cell walls.
Yes, two cell walls means two different cells. Okay, so when this is forming, it is not forming as one single unit. It is forming as two units. Okay, very closely packed. So this is called cell plate. And in between the two, there will be holes. These holes were Plasmodesmata, right? All the plant cells, now there must be a cell here also. There will be a cell here also. All the cell walls are connected to each other through holes in between them. Because they are so rigid, they are not going to allow some things or the other like plasma membrane. So they have small holes called Plasmodesmata. Is it clear?
Yes.
This makes it more clear how are they formed. Okay. This is the most, yes. So this is the cell plate which forms in the center. Rest about sometimes an exception. And also, let me tell you about exceptions. there are some cells called multi-nucleated cells. How are they produced? After studying the whole M phase and the interphase and the whole cell cycle, now can you tell me how do multi-nucleated cells are formed? Logically.
Sir, I think they will have 2 nucleus.
Exactly. Multi means many. Nucleated means many nucleus present in the same cell. So it is very logical that in these cells, karyokinesis will happen, but cytokinesis will not happen. Right? Yes or no? So if you do karyokinesis, you complete all prophase, metaphase, anaphase, telophase, and by the end there are 2 nucleus produced, but you do not do cytokinesis. Then it will be a single cell with 2 nucleus and it will be multi-nucleated. Right? So write down. In some organisms where in some organisms or cells where karyokinesis, nuclear division, karyokinesis is not followed by cytokinesis. Is not followed by cytokinesis. Give rise to a multi-nucleated condition. A multi-nucleated condition, and this condition is known as a syncytium. Syn means together. Syn always means together. Maha, we just studied about a syn word, right? Remember syngamy?
Yes.
In slime molds. So the syngamy, there the syn means together. So a syncytium means a cell which should actually be 2 cells, but is just 1 cell. It's living together. Continuous. Okay,
These cells
continuum.
have cytoplasm?
No, they will have cytoplasm. Lots of cytoplasm. Example is, how many of you like coconut water? Anyone likes coconut water? Yeah.
Me.
Have you ever thought what is a coconut water? How do water comes inside that coconut? Who fills it inside? Where does it comes from? It's all the cytoplasm which is present in a syncytium, not separated by cells. So these are millions of cells living together with their shared cytoplasm and shared nucleus. So what you are drinking is all the cytoplasm and the nucleus present in that water. So it's called a endosperm. It is an example of a syncytium where only karyokinesis is happening. So the whole coconut is actually like one big cell which kept on dividing but did not do cytokinesis, only did karyokinesis. Okay, so over time the liquid component kept on increasing because every time a cell divides, it synthesizes new cytoplasm. So it kept on synthesizing and dividing the nucleus. So if you take a glass full of coconut water, it just has free nucleus. So if you see under the microscope, there'll be millions and billions of free nucleus containing DNA that you are drinking. That's called a DNA juice. Ever thought of it? Aisha, don't cry, no? Why? It's good for health. It's protein. It's protein and it's nucleic acid. So that's why coconut water is very healthy and it's suggested for patients in any condition because it provides you with vital vitamins, amino acids, nutrients, carbohydrate, everything. So it is example, write down. Liquid endosperm in coconut is an example of syncytium. Liquid endosperm. And each nucleus there is 3N, is a triploid nucleus. So liquid endosperm in coconut, commonly known as coconut water. This is not the most bizarre thing, you know. I just don't want to discourage you from many other things. But I hope I have not spoiled coconut water for you. Aisha's heart is broken. Aisha, you'll stop drinking what? You'll stop drinking coconut water from now or what? Please don't do that. It's very, if you see that way, no, you will not be able to eat anything, because everything biologically is something which, if you extrapolate on a human level, it's gross. For example, curd. You like curd? What is a curd? A curd is just a colony of bacteria, millions of bacteria producing something that binds all the protein in the milk together and makes it curd. So you are eating all those bacteria very happily, but they are good bacteria. They go in your, Aisha is going to leave a lot of things now. I should stop. Don't worry, Aisha, they are good bacteria. And just to encourage you, have you heard of someone when you will become doctors, when someone undergoes loose motions or dysentery or diarrhea, these kind of issues, then we always tell that person to drink more and more liquid. And after you start healing, to get some probiotic drink, eat curd or get some probiotic drink, right? Yakult or something like that. You know why? Any idea? Maha?
No, sir.
No.
Because during a loose motion condition, we lose lots and lots of good bacteria from our guts. So our guts, our small and the large intestine, especially large intestine, hosts millions of bacteria. There are millions of good bacteria living in your intestine all the time, in all our intestines. That is called gut flora and gut fauna. So gut flora and fauna is very important because they make certain things for us which we cannot make for ourselves. You know, like certain vitamins and enzymes are produced by these good bacteria in our body. So during a loose motion, we lose these bacteria, like they are flushed out, because our stool is not properly dehydrated and it's not, it is just removed with lots of water. So these bacteria are just washed out. And then we become very low with these vital things. So we have to wait, drink liquid, and we have to wait for this bacterial colony to form again. And probiotic drinks help in that, especially curd and other Yakult-like products, because they have good bacteria in them. So we are eating bacteria, you know, yes, we are eating free nucleus of the coconut. And if I tell you about honey, honey is the most gross but very delicious thing. You know what is a honey technically? What honey is? How many of you like honey? Tell me. Aisha, Iram, Maha. Aisha, do you like honey?
Yes, yes.
Is it delicious? Maha also likes honey.
Yes.
You know what is a honey? It is the vomiting of a honeybee.
Yeah,
You know that, Iram, already?
I heard that it's spitting off the honeybee.
Yes. So honeybees, what they do, they go to flowers to collect the nectar, but they do not carry their pouches or backpacks with them, no? So the only place where they can collect that sweet substance is in their stomachs. So they suck on the nectar of the flowers, put it in their stomachs. When they reach the beehive, they regurgitate everything in those small compartments. And that is where honey is semi-digested already. And when you eat it, it gives you a lot of nutrition because you don't have to do much of hard work on that. And it's fructose, pure fructose, because flower nectars produce fructose. So it's called bee vomit actually. Technically, it's the vomiting of the honeybee, And honeybee is doing a good thing for you. She is collecting, honeybee is collecting all that nectar, putting it in the hive and semi-digesting it. So, you know, everything we get from living systems has to depend on living systems to survive. Okay, so that's about the cell division. I can understand you, Aisha. But don't worry, over time you will realize that everything you eat and you rely on is coming from a living system. Okay, because we cannot do chemosyn. We are not chemotrophic. We are heterotrophic. Okay, now coming to significance. Now this is one question. Write down. Question asked, what is the significance of mitosis? So we have studied mitosis till now. What is the significance of this process? In the end, significance of mitosis. Any idea? We know that from mitosis we can make equational. It is equational division. So we have discussed it already. All the repair thing that you do, growth, you know, everything that you do for sustenance, growth, and cell repair is done by mitosis. So write down. I've already told you that in animals, only diploid cells do mitosis. So that is the first point. In animals, only diploid cells do mitosis. Not do, undergo is a good word. In animals, only diploid cells undergo mitosis, whereas in plants and also some insects, and the insects I'm talking about is honeybee only. Honeybee, ants, etc. Haploid cells also undergo, haploid cells and diploid cells both can undergo mitosis. Remember these two examples, honeybee and ants. In these organisms, haploid cells also undergo mitosis. Second significance is the genetic component, because nothing is changed in mitosis. So the genetic component of the daughter cells remains same as the mother cell. It is just a process of copying. It remains same or identical. You can also use the word identical. Third, growth happens due to mitosis. Cell repair, tissue repair, all happens due to mitosis. And replacement of cells. You know, every time you eat something, you are scratching the outermost lining of your food pipe small intestine, large intestine every time, no matter howsoever soft food you eat. A lot of cells are just getting scratched off. Constantly they need replacement. You know that? So that is all done by mitosis. And it is done very fast. So have you ever, any one of you, by mistake, while eating or chewing the food, you accidentally cut your own inner lining of the cheek with your teeth? Has it happened? Maha? Yes.
Yes.
Okay. So, but how long does that wound stays? If you get a similar kind of cut on your skin outside, like on your finger or on your hand, versus in your cheek, which heals faster?
Cheeks.
Yeah. Have you ever thought of how and why? Because that is the evolutionary mechanism. See, every time we eat earlier, now we can cook food, right? So our dependency on hard food has gone down as a species. Humans do not eat very hard food these days. We can cook the food, we can make it soft, or whatever is not soft, we can blend it in a blender or a mixer. So we can do that. But other animals in the wild, or when our ancestors used to live in the wild, we were eating all sorts of things, right? Something which can create a cut in the lip or in the inner. And when we swallow things, also the inner lining also is damaged a little bit. So our gut and our mouth lining, especially our mouth inner cheek lining, is made up of epithelial cells, and they divide very fast. That's why if you cut it by mistake, it heals very fast. Sometimes between two meals, you will feel that, okay, not a problem, right? I can again chew. Yes or no? It just happens in frame of hours, correct? Whereas similar kind of wound on the palm or anywhere outside may take days. Yes? Clear?
Yes.
Yes.
Now, this is one reason. So, you know that plants can grow for their whole life. They actually grow till their whole life. Animals stop growing after one particular age. So humans grow to a maximum height, width, and everything, organ shape, size. Our hands, legs, limbs, our height reaches a pinnacle and then it stops. Yes? But plants never stop. They keep growing continuously for their whole life. That is because the mitotic division keeps happening in their meristematic tissues. Okay, so write down the continuous growth of plants throughout their life. The continuous, that's why we have to cut the bush after every some time, or trees, we have to trim their branches. The continuous growth of plants throughout their life is due to mitotic divisions in their meristematic tissues. Mitotic divisions in their meristematic tissues. Is it clear?
Yes, sir.
And if you have not studied, let me tell you, the meristematic tissues are of two types, because somewhere down the line you will study it. Just because we have started this thing, let's discuss it further. One type is called apical. This is required for the apical meristem. This is responsible for the height of the plant. The plant keeps, so plant grows in two directions. If you see a tree growing, the tree gets wider, thickened over time also, like the bark also becomes thick and the height also goes up, right? Yes or no? And both keeps happening throughout. So one is called apical meristem and the other is called lateral meristem. Okay. Apical meristem is for the height of the plant and lateral meristem is for the width or the girth of plant. Okay. And also for the branching. Is it clear, everyone?
Yes, sir.
Okay, very good. Now, you have studied mitosis, the whole cell cycle of the mitosis from the beginning till the end. Now we have to the next part of this chapter, the other half of this chapter is meiosis. Now meiosis we will do together. It's the same thing, but the only difference is it happens twice. In one cycle, there are two division cycles. That's the only difference in meiosis and mitosis. It's reductional division. So I'll explain you that. But before that, take 3-4 minutes and go through the whole mitosis once, all your notes. Ask me if you have any doubts, and then meiosis we will finish in the next half an hour together. You need to do two things. All three of you have NCERT with you?
Yes, sir.
You have NCERT? Your NCERT with you? The textbook?
Yes.
NCERT. Aisha?
Yes, sir.
Yes. Iram?
Yes.
Very good. Keep it handy. But before that, go through everything I have told you, because the same thing is repeated, but twice in meiosis, and I'll tell you what is the difference. So go through everything from the beginning till the end. Take 4 minutes. Okay. I'll take a quick water break in that. You do the revision. Okay, Iram? Then we'll start meiosis. Okay, everyone? Someone? You started revising, yeah? Say yes or no.
Yes.
Okay,
Yes.
do it. Do it, do it. Is it done, everyone? Is it not working? Am I audible?
Yes, sir.
Am I visible also?
No.
Oh, there's some issue. Okay, I'm audible. Okay, am I audible, people? Aisha, Iram?
Yes.
Okay, very well. Any doubts?
So yeah.
Yeah, please tell me.
So in metaphase, there's a point called. It is the stage at which chromosomes are seen. Like morphologically, we can see chromosomes.
Mm-hmm. Yeah, see,
And why is
okay.
it?
Okay, so this is the question. Like
Yeah.
seen, see, okay. See, chromosomes can be seen in prophase also. Okay. And very clearly it can be seen composed of two chromatids attached together at the centromere. Okay. In metaphase, it is seen properly at the center plate, metaphase plate. So it becomes easier to see all the chromosome aligned together, because in prophase, it can be seen because it's compacting. So late prophase or early metaphase, when they have not aligned on the plate, but nuclear membrane has also dissolved. So that is late prophase, or you can call it early or onset of metaphase. As I told you, there is no clear-cut checkpoint like when can you say it metaphase and when can you not say it late prophase. It's like the moment nuclear membrane disappears and all the chromosomes are in the cell, from there onwards, we start calling it the beginning of metaphase. So that's why they can be better stained and visualized in a metaphase, early metaphase. But they can also be seen in prophase. They can also be seen in anaphase, but during anaphase, till they are moving, they are seen. The moment they reach at the poles, they disappear. They decondense. Okay, this is why. Whenever we have to see a chromosome, you know, how do we see a chromosome? Chromosome are just colorless. You cannot see it inside a cell. But we put a stain, which is positive, a positive protein. DNA is negatively charged. You know that? DNA is called deoxyribonucleic acid. It is an acidic molecule. So acids have negative charge on them. If you put something, a stain which can bind with this DNA, you can see the chromosomes. Okay, so chromosome staining is a very normal thing. You put a stain in the cells and the chromosomes get marked. So that you can very clearly see during the metaphase. Okay, late prophase also and early anaphase also, but majorly at metaphase because every one is at the aligned at the center. So it's easier to visualize. Okay, that's why books say it. Okay, Iram?
Yes, sir. And also the ploidy of these chromosomes would be different at all the levels, right? Prophase, metaphase, anaphase.
Ploidy. Ploidy in mitosis is going to be same every time. It is an equational division. So the ploidy remains same. That is what is the concept of mitosis, right?
Yeah.
Ploidy only changes in the S phase. Before they enter into the M phase, S phase, the whole DNA genome, whole genome gets doubled. So whatever was 2 copies, I told you, no, if there is 5 kgs of substance, it becomes 10 kgs in the S phase. There, for the time being, ploidy becomes double. But when they enter into the cell cycle, prophase onwards, when they come out in the nucleus, so prophase, you can say yes, prophase will have twice the number of chromosomes. Then they all will line at the metaphase. Sorry, the ploidy will remain same, but it will have twice the number of genetic material. If I go to S phase, remember I told you that during the S phase, where is S phase? S phase, S phase, S phase, yes. So DNA replication happens, but check your notes. Have I dictated you this? That the content of the DNA doubles, but the number of chromosomes remains the same.
Yes, sir.
It's there in your notes? Yes? That's a very crucial point. Just remember that. So DNA content gets doubled, but the number of chromosomes remains same because every chromosomes makes 2 sister chromatids. Okay? Clear, everyone?
Yes.
Now let's go to meiosis and see what you have understood. Now meiosis is just slightly complicated, but the concepts are same. If you have understood mitosis, we just have to know that meiosis. So let me make this demarcation and start with meiosis here. Now about meiosis, in one cycle, like one meiotic cycle. So first write down this. From one diploid mother cell, 4 haploid daughter cells are produced. This is the characteristic feature of meiosis. Okay. So if I draw it, so there is a mother cell. It has 2n, like 2 copy number of the genome. One copy came from the mother, the other copy came from the father. Yes? Understood? Now, in one cycle of meiosis, there are 2 mini cycles. So there are 2 cell cycles in one meiotic division cycle. Now, first is called meiosis 1, M1. And meiosis 1 is reductional. In this, 2 cells N and N are found. You understood? And then
Yes.
each cell undergoes one more cycle, which is called meiosis 2, in which again 2 cells N and N and N. Now we started with a diploid mother cell, and after meiosis 1 we got 2 haploid daughter cells, and after meiosis 1 we got 4 haploid daughter cells. So pay attention here. Okay. If we just look at this diagram now. We started with this one cell. If it was mitosis, there would have been only one cycle, and 2n will give rise to 2n and 2n. So this is meiosis. Let me draw mitosis here. So in mitosis, we start with 2n, and then there is only one cycle, and the daughter cells are also 2n and 2n, right? Mother cell, diploid, these are daughter cells. So this is simple. That's why it's called equational division. Here, the first M1 is reductional, right? So do you think DNA replication has happened here?
Yes.
Yes, Aisha? If DNA replication will have happened, this would have become 4n. And upon dividing, it will be 2n and 2n. So DNA replication happened here. During this stage, DNA replication, or let me call you DNA duplication happened. Yes or no? Otherwise, it is not possible from 2n. If you have 2 rupees and you ended up giving 2 rupees to 2 people, it clearly tells that you brought 2 more rupees from somewhere, right? It's not possible to distribute 4 rupees from 2 rupees. So if you do the total, it is 2n plus 2n, 4n amount of DNA. It had already just 2n DNA. So DNA replication happened here, right? In meiosis 1, did you think DNA replication happened?
No.
No. Yes. What about this meiosis 2? In meiosis 2, do you think DNA replication happened?
Yes.
It was yes.
Yes.
Yes, because it was N and it ended up giving N to 2 daughter cells. So meiosis 2 is actually, it mimics mitosis. Yes or no?
Yes.
And meiosis 1 is reductional in nature. Yes?
Yes.
And meiosis 2 is equational in nature. You can see it is equal. So there are 2 cycles, 2 cell division cycles and nuclear division cycles, but only 1 DNA replication cycle in meiosis. So there are some interesting differences between meiosis and mitosis, but the fundamental similarity is same. So first write down some things about meiosis. Write down. Gametes for sexual reproduction. You know what are gametes?
Yes.
Yes. What are gametes? Sperms
Sperm and ovum.
and ovum. So gametes for sexual reproduction are formed through meiosis. Are formed through meiosis. And gametes are haploid cells. Okay. Meiosis is very, very important for sexual reproduction, because if meiosis does not happen, then from a diploid cell, we will not be able to make haploid gametes. And if we are not able to make haploid gametes, then the problem of maintaining our genome, our ploidy, will be there. For example, a male human is diploid, right? And a female is also diploid. Because of meiosis, a male can make a sperm, which is haploid. And a female can make a ovum, which is haploid, right? And this is happening because of meiosis. And then both of them fuse together, they form a zygote, which is 2n. And then this zygote becomes a new human, right? Which is again 2n. Yes or no? Then this new human will again go, either it will be a male or a female, and it will make again sperms. So after generations, we can conserve our ploidy. Do you understand? Our
Yes.
parents are diploid, but we are also diploid. Our grandparents were also diploid. Our great grandparents were also diploid. This is only possible because we have a mechanism, a different kind of cell division, which we only use for making gametes. For rest, everything in our body, for growth, development, repair, replacement, wound healing, recovering from injury, we do mitosis. But just for gamete formation, we do meiosis. If we could not have done meiosis, then this was the problem. problem would have been, let's
The
see here.
ploidy will increase, right?
Yes. So there will be a male 2n and a female 2n. But since they cannot do meiosis, they will only do mitosis. So their gametes will also be 2n, right? Because mitosis is equational. Yes. And when they will fuse, when they will reproduce, the zygote will be (4n). You understand? People? Say yes or no.
Yes, sir.
And the problem is this 4n then again will, if survive, so animals cannot tolerate polyploidy. So this zygote, this is all hypothetical, but if meiosis was not present in biology, then we would have ended up this problem. Now this person grew up to become another, let's say, male, which is 4n. Now it can either find a female who's 2n or 4n or 8n, depends, right? So even if he finds a female 2n, again they cannot do meiosis, so the gametes will be 4n and her gametes will be 2n. They will fuse together. The next generation will be 6n. You understand? It cannot happen. We are humans. We are all humans. We have all same genes, same number of copies of the genes, but it will just keep on increasing generation by generation. So this is the problem that will happen if you do not have meiosis as a cell division mechanism to make gametes. Understood clearly?
Yes, sir.
It will be impossible to maintain ploidy across generations in the absence of, you can write down this one sentence, in the absence of meiosis for gamete formation, in the absence of meiosis for gamete formation, it will be impossible to maintain ploidy. It will be impossible to maintain ploidy through generations for any species. Done? Okay. Maha, welcome. Maha has started using emojis. Nice. Good. This batch is emoji batch. I use this one and for me it's slightly complicated to go to reactions and yes, see, I did it. Okay, write down the next important thing about meiosis. So gamete for sexual reproduction are formed through meiosis. Second, meiosis involves two cycles of karyokinesis and cytokinesis. Karyokinesis and cytokinesis. So nuclear division and cell division, right? So by now this term is in your memory, right everyone? Maha, Ira, Maisha?
Yes.
So meiosis involves two cycles of karyokinesis followed by cytokinesis, known as meiosis 1 and meiosis 2 respectively. And meiosis 2 respectively. You do not need to put respectively here. Meiosis 1 and meiosis 2. But it has only one cycle of DNA replication. Only a single cycle of DNA replication. Okay, and when does DNA replication, in which cycle does DNA replication happens? During the meiosis 2, right? Because meiosis 2 is like, meiosis 2 mimics mitosis, right? So just know this, meiosis 2 mimics mitosis only. Is this clear, everyone?
Yes. Sir.
Now, before I go, so this is, if you find it difficult, only meiosis 1 is difficult, and in fact, even in meiosis 1, only the prophase is difficult. So please come to page number 168 of your NCERT, please, quickly. 168 people. Did you see 168, 169 and 170?
Yes.
3 pages are for meiosis only. Do you see prophase 1? So because see, it's simple here also. Come to page 167. You can see that meiosis 1 has the same phases. Prophase, because everything is 1, so it is prophase 1, metaphase 1, anaphase 1, telophase 1. Then meiosis 2 starts. That is prophase 2, metaphase 2, anaphase 2, and telophase 2. Can you see that?
Yes.
Yeah. So I'm giving you a homework today for this week before you come to the next class. The same things happen, but prophase 1 is slightly longer in meiosis because every step of condensation of the DNA. Remember, in prophase 1 of mitosis, there were 2 things happening. One, the condensation of the chromatid condensation of the DNA in the nucleus, and movement of centrosome in the cytoplasm towards opposite poles. Yes or no?
Yes.
So
Yes.
movement of cytoplasm will happen here also, but with respect to the condensation of the DNA, it is more elaborate. So your homework is you have to read page number 168 to 170, these 3 pages. And make your notes and try to understand how it is same and different from mitosis. In the next class, when you will join, after the summary, I'll just explain this whole thing to you, and the chapter will be finished in the next class, and we'll see. So one more thing, if it is okay, so Iram and Aisha, 8 and 10, what else is coming in your exam in September? Which chapter?
Morphology and flowering plants and anatomy.
Biomolecules.
Biomolecules is for whom?
Yes.
No, that's not started, right?
That is coming.
Biomolecules is coming or not? And what is coming in? So Maha's syllabus includes chapter 1 and 2 also, right, Maha?
Yes.
And apart from that?
And I think chapter 3 as well.
3 as well. Okay.
Yeah.
So let's do one thing, because we have finished cell and cell cycle. Let's also quickly go to biomolecules, which is the in-between chapter, right? Shall we go to biomolecules next? And then after learning about cell and the molecules inside the cell, we'll come to chapter 1, 2, and 3. How about that?